Recall, parenchyma of kidney is made of renal cortex, which is the outer region of the kidney that has a lighter color, and renal pyramids. Renal pyramids refer to these cone-shaped structures that we see in renal medulla. That simply means that the nephrons, microscopic structures that filter blood and eventually produce urine, are located in the renal cortex and the renal pyramid. Here we have a closer view of a renal pyramid and the renal cortex close by, and we can see several nephrons located in the renal cortex and in renal pyramid. Each nephron is made of two distinct portions. The beginning of the nephron is referred to as renal corpuscle, and the long tube that each nephron has is called renal tubule. In this view, we can see two of the nephrons completely, and as you can see, even we opened the renal corpuscle of these two nephrons to see what we have in each renal corpuscle. So please keep in mind that in all the other renal corpuscles, if we open them, we can see exactly what we have here. Deep inside, we have a ball-shaped capillary, and wrapping around the capillary, we have a capsule. So renal corpuscle is made of two structures. The capillary that sit deep inside, we name that capillary glomerulus, and the capsule that encircles the glomerulus. We refer to this capsule as either glomerular capsule or Bowman's capsule. And we have a closer view of a renal corpuscle. That would be the capillary that sit deep inside glomerulus, and this would be the Bowman's capsule. As you notice, Bowman's capsule has an outer wall, we call this parietal layer of the Bowman's capsule, and an inner wall. Inner wall is the wall of the Bowman's capsule that wraps around the capillary. In both parietal layer and the inner wall, which is referred to as visceral layer, we have simple squamous epithelium, one layer of flat cell. When we look at the simple squamous epithelium of parietal layer, we see that it's just a normal, usual, simple squamous epithelium. However, when we look at the flat cells that we have in visceral layer, we notice that these are very large flat cells, and we name these modified large flat cells podocytes, and each podocyte has some projections that we refer to these projections as pedicels. So in the visceral layer of Bowman's capsule, we can find podocytes, the cells that have projections named pedicels. And between these projections, we can find a very thin layer made of protein. We refer to that layer of protein that is located between the projections of these cells as slit membrane. In reality, these podocytes wrap around the entire glomerulus, the entire capillary. However, please note in this model, they have removed podocytes from half of the model to expose the capillary. As soon as we look at the capillary, the glomerulus, we notice that the endothelial cells have pores, fenestrations. So glomerulus is a fenestrated capillary, which we know having these fenestrations allow the glomerulus to increase filtration. So when it comes to filtration, which would be pushing water and solute from glomerulus into the Bowman's capsule, water and solute should pass through the fenestrations of these endothelial cells. Then they must pass through the basement membrane of the endothelium. And last but not least, water and solute must pass through the slit membrane, that layer of protein that we find between the pedicels. So in short, filtration takes place in renal corpuscle between glomerulus and the Bowman's capsule. And recall that the blood vessel that brings blood into the glomerulus is an arteriole, and the blood vessel that takes blood out of the glomerulus is another arteriole. So that's the unique characteristic of glomerulus that sits between two arterioles. The arteriole that sends blood into the glomerulus is afferent arteriole, and the arteriole that drains blood from glomerulus is referred to as efferent arteriole. So when I go back to this model, following what we learned about the blood vessels that supply the kidney, we see that from interlobular arteries, or we name them cortical radiate arteries, we get afferent arteriole that send blood into the glomerulus, and then efferent arteriole takes blood out of the glomerulus, and eventually efferent arteriole forms those capillaries that wrap around the tubules. So one more time, from cortical radiate artery, we get afferent arteriole, and we know inside the Bowman's capsule we have the glomerulus. After glomerulus we have efferent arteriole, and we see efferent arteriole forms the peritubular capillaries that we have around the renal tubule. And recall that when we get inside the renal tubule, two physiological processes are going to take place. One would be reabsorption, which would be sending substances that we need, solutes and water that we need, from renal tubule back into peritubular capillary, and the toxic substances and waste that still we have in blood from peritubular capillaries move into the renal tubule. That would be secretion. In short, as soon as filtration takes place and we get inside the Bowman's capsule, we refer to the fluid that we have in Bowman's capsule as filtrate, and then filtrate from Bowman's capsule enters the renal tubule. Here we have a very short section of the beginning of renal tubule. The beginning of the renal tubule is called proximal convoluted tubule, simply because it's a coiled tube and it is attached to the Bowman's capsule. One important thing about proximal convoluted tubule is that here we have the most reabsorption. For example, if you focus on a molecule such as glucose, glucose from glomerulus is filtered into the Bowman's capsule. So inside the filtrate we find glucose. However, we know glucose is a nutrient. We do not wish to excrete glucose out of our body in a healthy individual. So when glucose molecules in the filtrate get inside the proximal convoluted tubule, we have 100% reabsorption of glucose, means all the glucose molecules from proximal convoluted tubule go back into the blood that flows in peritubular capillaries. So in a way we can say that proximal convoluted tubule has the highest percentage of reabsorption of substances that we need to keep. After proximal convoluted tubule, we refer to the U-shaped portion of the renal tubule as nephron loop or loop of Henle. But please note, the first section of nephron loop goes down and penetrates the renal pyramid. We refer to this section as descending limb of the nephron loop, and the rest of nephron loop goes back up to enter the renal cortex again. We refer to this part of nephron loop as ascending limb of nephron loop. So nephron loop has descending limb and ascending limb. After nephron loop, we have another coiled tube, but please note that this coiled tube is not attached to Bowman's capsule. We refer to this one as distal convoluted tubule, and we name the first part of the distal convoluted tubule as early distal convoluted tubule, and the rest, the end of distal convoluted tubule, is referred to as late distal convoluted tubule. Then we see distal convoluted tubule of several nephrons, like this one, and distal convoluted tubule of the other nephron that we have here, drain filtrate into a pathway that is called collecting duct. So collecting duct receives filtrate from distal convoluted tubules of several nephrons, and when we follow collecting duct, we see that it takes filtrate eventually into papillary duct. And as you see, papillary duct ends exactly at apex of the renal pyramid, or we refer to the apex as renal papilla. that would be the papillary duct. And when we follow the papillary duct, we end exactly at renal papilla. And on the other side of renal papilla, we have these cup-shaped spaces. We refer to these cup-shaped spaces as minor calyces. And we know as long as filtrate flows through the renal tubule, collecting duct, papillary duct, reabsorption is possible. However, as soon as we get inside the minor calyces, we have urothelium here, which does not allow any reabsorption to take place. So what gets inside the minor calyces must be excreted out of our body. That's why as soon as we get inside the minor calyces, we refer to the fluid as urine. If you follow any of these nephrons, you see that the arrangement of these sections is the same. For example, if I analyze the other nephron that I see on the right side, first I have renal corpuscle, which is made of the capillary inside glomerulus and the Bowman's capsule encircling it. Then the first part of the renal tubule is proximal convoluted tubule. Then I get to descending limb of nephron loop, ascending limb of nephron loop, early distal convoluted tubule, late distal convoluted tubule, collecting duct, and papillary duct. And recall that around all different sections of renal tubule, we have these peritubular capillaries. We have two groups of nephrons in our kidneys. About 80 to 85% of the nephrons in kidneys are referred to as cortical nephrons, and about 15 to 20% are juxtamedullary nephrons. When we focus on the renal corpuscle of cortical nephrons, we see that the renal corpuscle sits in the outer part of the renal cortex. However, the renal corpuscle of juxtamedullary nephrons sits in the deeper part of the renal cortex. The next difference is the nephron loop of cortical nephrons is very short, and the nephron loop extend only into the outer part of the renal medulla. However, the juxtamedullary nephrons have very long nephron loops that extend into very deep part of the renal medulla. And another thing to note here is that juxtamedullary nephrons have significant role in either diluting urine or concentrating urine, which in separate videos we'll learn about these processes. One interesting fact to know about juxtamedullary nephrons, aside from their very long nephron loop, is the anatomy of their ascending limb. When we follow the ascending limb of these nephrons, we see that the first section of the ascending limb has a very narrow diameter. We refer to it as thin ascending limb. And the rest of the ascending limb has a wider diameter. We refer to this as thick ascending limb. And later we'll learn that there are differences between the physiology of thin versus thick ascending limb. For example, in thin ascending limb, we have some reabsorption of water. Recall that simply means we're sending water from thin ascending back into these peritubular capillaries. However, when we analyze the cells that line the thick ascending, we see that mostly they are lacking the water channels that we need for reabsorption of water. As a summary, we know that the interlobar arteries are the ones that bring blood into arcuate arteries, as I explained in the video about blood supply of kidneys. And then from arcuate arteries, we get into cortical radiate arteries. And as I mentioned in this video, from cortical radiate arteries, we get the afferent arterioles that send blood into the glomerulus. From glomerulus, water and solutes are pushed into the glomerular capsule. That would be filtration. And then blood leaves the glomerulus and gets inside the efferent arteriole. So here I have the efferent arterioles, and when I follow them, I see that they form the peritubular capillaries. When I follow the peritubular capillaries, I see that eventually from these peritubular capillaries, blood goes into cortical radiate veins that empty blood into arcuate veins, and then from arcuate veins, blood goes into the interlobar veins. But please note that between peritubular capillaries and the different sections of renal tubule, we have reabsorption and secretion happening. Reabsorption, you must go from the tubule into blood, and secretion from peritubular capillary, you send the toxic substances into the renal tubule to eventually eliminate them. And when we follow the interlobar veins, we know that eventually they send this filtered blood into the renal vein that empty blood into the inferior vena cava.